Imagine you are moving into a new apartment, and it's on the top floor of this very old, very cramped building.
Oh, I hate moving.
Right. It's the worst. And to make it worse, you have a fully assembled grand piano.
Okay.
And the only way up is this incredibly narrow, winding staircase. So you push and you shove and you wedge it around the corners.
Scraping the walls all the way up.
Exactly. By the time you finally get to the living room, the drywall is scraped, the banister is loose, and the piano itself is just, scratched and dented.
Oh.
You had to cause a measurable amount of damage just to get to your destination.
That is a very visceral image. And honestly, it perfectly captures the biological paradox we are exploring today.
Yeah, and welcome to today's deep dive, by the way. I'm so glad you're joining us for this one.
Because we have this ingrained assumption that healthy biology, especially brain development, is this pristine, error-free process.
Right.
We think of it like architectural construction. Very clean, very precise. But sometimes just the basic mechanics of development are, well, they're inherently violent.
Oh, inherently violent. And that brings us to the core mission of today's deep dive. We are looking at a really mind-bending 2026 paper from the journal Nature.
Yeah, a fantastic paper.
The study is by Zhang and colleagues, and it's titled Confined Migration Induces Non-Lethal DNA Damage in Developing Neurons.
Which is a mouthful.
It is. But the central terrifying idea here is that to become a functioning brain cell, a newborn neuron must literally shatter its own DNA just to squeeze into its final position in the brain.
Yeah, and the context here is really what makes it so revolutionary, because we've known for decades that neurons accumulate DNA damage over a lifetime. If you think about oxidative stress or environmental toxins, just the general wear and tear of aging.
Right.
But what this research team uncovered is completely different. They demonstrated that the simple mechanical act of normal physiological migration, literally just the embryonic brain wiring itself up, causes massive DNA double-strand breaks.
Okay, so we are talking about the very first moments of life. The paper focuses specifically on cerebellar granule neurons in mice, right?
Yeah, exactly. Often just called CGNs.
CDNs. Okay, I'm trying to picture the actual journey these cells are taking. Where are they starting and where are they going?
So during the first 3 weeks of a mouse's life, these newborn cells are generated on the outermost layer of the cerebellum.
Okay.
But to properly wire the neural circuits, they can't just stay on the surface. They have to migrate radially inward, diving deep into the internal layers of the brain.
So they're moving into the core.
Right. And to do this, they have to drag their massive nucleus, the central vault containing all of their DNA, through incredibly dense, crowded, tightly packed brain tissue.
Okay, so they really are pushing the grand piano up the stairs.
Exactly.
But how do you even observe that kind of microscopic damage in a living cell? I mean, it's not like you can just look at a neuron and see a crack in it.
No, you can't. You need a way to make the invisible visible. So the researchers used a set of fluorescent markers designed to light up specific biological events in real time.
Okay, what kind of markers?
Well, the key ones are called gamma H2AX and 53BP1.
Gamma H2AX and 53BP1.
Got it. Yeah, without getting too bogged down in the chemistry, you can just think of those markers as a flashing red siren.
A siren.
Right. In biology, when a DNA double-strand break occurs, the cell immediately tags the site of the break. The researchers basically engineered these tags to glow.
Oh, that's clever.
Yeah. And 53BP1 essentially illuminates the emergency repair crew arriving at the scene.
So you have the red siren going off and the ambulance arriving, and it's all glowing under a microscope.
Precisely. But they also had to be sure of what they were looking at. So they used two other markers, one called Chi67.
Okay, what's that one, dear?
Chi67 marks dividing cells, and then another one called P27-KIP1.
P27-KIP1.
Exactly. And that one marks mature postmitotic cells, cells that are done dividing.
Ah, so they wanted to make sure these were mature cells that were just traveling, not cells that were just splitting in half.
Exactly. And when they looked at the developing brains, they saw these traveling post-division cells completely lighting up with those red sirens. The gamma H2AX and 53BP1 were glowing everywhere.
I have to stop you there, though, because, correlation isn't causation.
Fair point.
Is it really just the tight squeeze causing those breaks? I mean, if a cell is moving through a dense neighborhood of other cells, it's being bombarded by chemical signals.
Sure.
Could it be like a growth hormone or just some programmed stage of development that's triggering the DNA to alter?
Yeah, and that was the exact hurdle the researchers had to clear. To prove the damage was purely mechanical, they stripped away the brain entirely and built an artificial obstacle course in a petri dish.
An obstacle course for cells?
Yeah, They created tiny microfabricated channels for the neurons to crawl through. Some of the channels were 8 micrometers wide,
Okay.
which for a neuron is a very permissive, comfortable hallway. Plenty of room.
But the others?
The others were just 3 micrometers wide, a highly confined, crushing squeeze.
Wow. And I'm assuming the 3 micrometer squeeze is where the sirens started going off.
Emphatically, yes. The neurons migrating through the comfortable 8 micrometer channels, they're perfectly fine.
No sirens.
No sirens. But in the 3 micrometer channels, the central nucleus had to severely deform to fit through the gap. And the exact moment they wedged themselves in, there were massive spikes in the damage and repair signals.
Wow.
It stripped away all the other variables. It wasn't chemical signaling. It was the physical, mechanical squish directly breaking the DNA strands.
Okay, let's take a quick pause here because we really want to make sure we're absorbing this step by step. So let's do a quick recap for you listening.
Good idea. Let's lock it in.
Okay, bullet point 1. Newborn neurons have to migrate through really dense brain tissue to get to their final spot.
Right. Bullet point 2. When they squeeze through tight 3 micrometer spaces, it physically stresses the nucleus.
And bullet point 3. That mechanical stress directly results in transient DNA double-strand breaks. The sirens go off.
Spot on.
So here's a question for you to chew on while we move to the next part. Have you ever considered that the very act of growing a healthy brain involves inherently destructive physical forces?
It really reframes how we think about the fragility of the brain, doesn't it?
It totally does. But it also introduces a massive mechanical puzzle because, well, cancer cells migrate too, right?
They absolutely do.
Tumors spread by squeezing through tight tissues all over the body. Do they take damage the exact same way?
They do take damage, but they handle the physical crush very differently. When cancer cells squeeze through tight spaces like that, their nucleus suffers what we call nuclear envelope rupture.
Meaning the outer shell just pops.
Yes, the membrane literally tears open. Think about a water balloon being forced through a chain link fence.
Right. If you push hard enough, the rubber stretches, thins out, and eventually snaps. The balloon bursts.
Exactly.
So for a cancer cell, the envelope bursts, and I'd imagine that's a complete disaster. All the internal DNA repair factors leak out into the cellular fluid, and destructive enzymes from outside the nucleus rush in, just chewing up the exposed DNA. The cancer cell has to frantically patch the hole.
Yeah, this is exactly what happens. And the researchers proved this happens in cancer cells, but not in neurons, by using a really clever alarm system.
Another marker.
Yeah. It's called GFP-CGAS.
GFP-CGAS.
Right. And this marker only lights up if the nuclear envelope tears.
Okay.
So they put a famous line of human cancer cells, HeLa cells, through the 3 micrometer obstacle course, and the alarm lit up like a Christmas tree.
Oh, wow.
Their envelopes were tearing left and right.
Yeah.
But when they forced the migrating newborn neurons through the exact same microscopic squeeze, 0 rupture.
None.
The alarm stayed completely dark.
Wait, why is the neuron so much better at squeezing than a heavily mutated cancer cell?
The secret lies in a structural protein called lamin.
Laminae.
You can think of lamin A as the steel rebar in a concrete wall.
Yeah.
It gives the nucleus stiffness and rigidity.
Okay, so more laminae means a stiffer nucleus.
Exactly. And neurons, it turns out, express extraordinarily low levels of lamin A because they lack this rigid structural rebar. Their nuclei are incredibly soft and deformable.
Oh, I see.
They can squish and stretch to dramatic lengths without snapping.
So it's less like a water balloon and much more like a blob of silly putty passing through the fence. It just morphs to fit the shape.
There's a perfect analogy.
But wait, hold on. That creates a massive logical problem.
How so?
If the neuron's protective shell never breaks and there are no destructive enzymes leaking in from the outside, how is the DNA inside getting smashed?
Whoa.
It's like finding a sliced apple inside an entirely uncut, pristine peel. What is doing the cutting?
Okay, let's do another quick recap of this section to frame that mystery.
Good call. Let's do it.
So, bullet 1. Unlike cancer cells, migrating neurons do not rupture their nuclear envelope.
Bullet 2. They survive the squeeze because they have very low laminae levels, giving them a soft, ultra-flexible nucleus.
And bullet 3. The DNA breaks are happening inside an entirely intact nuclear shell.
Right. Which brings us to the question for you listening. If the protective shell never breaks, what phantom force is slicing the DNA inside?
Right. To find that phantom force, the researchers had to look strictly inward. The damage wasn't coming from outside the nucleus, it was coming from an enzyme that lives inside.
Okay, an inside job.
An inside job. It's called topoisomerase-IIβ, or just TOP2 beta for short.
TOP2 beta. Let's make this tangible. I'm looking at my notes here, and it says it acts essentially as a highly specialized pair of molecular scissors.
Yeah, that's the best way to visualize it. But to understand why the cell is spontaneously deploying scissors on itself, you have to understand torsional stress.
Torsional stress, like twisting.
Exactly. DNA is a double helix, right? A massively long, twisted ladder. If you unspooled the DNA in a single human cell, it would be about 2 meters long.
2 meters in a single microscopic cell.
Yeah, it's packed in there incredibly tight. Now, when a cell compresses its nucleus to wedge through a 3-micrometer pore, that 2 meters of DNA gets intensely crushed and twisted upon itself. It builds up enormous physical tension.
Oh, I see.
If you take a rubber band and twist it over and over, eventually it starts bunching up into tight, uncontrollable knots. DNA does the exact same thing under pressure.
And if you don't relieve that mechanical tension, the DNA ladder will just snap randomly across the sequence, which would be catastrophic.
It would be.
So the scissors step in to prevent a worse disaster.
Exactly. TOP2's normal, healthy, everyday job is to find those overwound, dangerous knots of DNA, intentionally cut both strands to relieve the twisting tension, pass another strand of DNA through the temporary gap to untangle the knot, and then perfectly glue the cut ends back together.
That is wild.
It's an incredibly elegant, controlled way to manage physical stress.
So the cell is purposefully breaking its own DNA to keep it from snapping blindly. But hang on, if it cuts the DNA and then glues it right back together immediately, why were those flashing red sirens going off for so long under the microscope earlier?
Here's where the biology gets terrifying. Under the extreme abnormal pressure of the tight squeeze, the molecular scissors get physically jammed. Yeah. TOP2 makes the initial cut, but because the physical distortion of the whole area is so severe from the 3-micrometer squeeze, it fails to glue the strands back together. It becomes chemically locked onto the broken DNA ends. They call it a TOP2 cleavage complex, or TOP2CC.
Oh man, it's like you're untangling a massive knot of extension cords behind your TV, and you cut a wire to free it up, but your metal scissors get permanently wedged into the copper cable.
Exactly.
Now you have a severed wire and a pair of scissors jammed in the break, blocking you from taping it up.
That's exactly it. And the cell can't just pull the scissors out. They are chemically fused.
Wait, if the scissors are stuck and the DNA is severed, how does the cell not just immediately trigger its own death sequence? It seems like a biological dead end. I mean, a broken genome usually triggers a cell to just self-destruct for the safety of the organism.
That is exactly what you'd expect. But instead of dying, the neuron activates a cellular garbage disposal system. It sends specialized machinery, a proteasome, to literally dissolve the trapped TOP2 scissors.
Dissolve?
Yeah. Only after the jammed enzyme is completely degraded and chewed up are the broken DNA ends exposed so the cell's repair crew can finally access them.
That sounds like an absolute nightmare to prove in a lab. How do you prove a microscopic pair of scissors is getting stuck and then getting eaten?
Well, they used a brilliant pharmacological trick, a 1-2 punch. First, they introduced a drug called adiposide.
Etoposide.
Right. This drug specifically forces those TOP2 scissors to jam after they make a cut.
Okay.
Even with a tiny drop of this drug, the DNA breaks persisted significantly longer after the tight squeeze.
So that proves the scissors are involved.
Right. Second, they used a completely different drug called epoxomycin.
Epoxomycin, what does that do?
That one shuts down the proteasome, the garbage disposal system.
Ah.
And when they stopped the disposal, the broken DNA stayed permanently broken, proving that without the ability to dissolve the jammed scissors, repair was impossible.
Okay, let's do a recap on this whole sequence because that was a lot of mechanism.
Let's do it.
Bullet point 1. Mechanical pressure causes intense twisting, torsional stress on the DNA.
Bullet point 2. The TOP2 beta enzyme intentionally cuts the DNA to relieve tension, but under the crush, it gets trapped.
And bullet point 3. The cell has to use a garbage disposal to degrade the trapped enzyme, leaving a temporary double-strand break in the process.
Perfect.
So the question for you listening, how does a cell prioritize which parts of its genetic code are safe to cut when it's under that kind of immense physical pressure? Because you don't want to accidentally slice the page of the manual that tells your heart how to beat.
And that is the perfect pivot to the cell's defense strategy. Because, as you said, a double-strand break is arguably the most lethal type of damage a cell can face. The neuron needs a rapid cleanup crew, and it needs to be incredibly strategic about where it allows these cuts to happen in the first place.
The cleanup crew they identified is a repair pathway called non-homologous end joining, right? NHEJ,
That's the one.
which we've been calling the duct tape pathway.
Right, the duct tape pathway. It is heavily reliant on a specific enzyme called ligase-4, or LIG4, to seal the DNA back together.
LIG4.
Got it. Now, this pathway doesn't use a matching template to perfectly recreate the missing code. It just grabs the two broken, frayed ends and jams them back together as violently and quickly as possible.
Wow.
It's incredibly fast, but it is notoriously prone to making tiny errors, dropping a letter here, adding a letter there.
So if your only tool is error-prone duct tape, you definitely don't want this happening in your most critical genes. Where exactly in the massive genome are these breaks actually occurring?
To figure that out, the researchers used an advanced sequencing technique called e-TISEC.
E&DSEC, meaning they mapped the ends of the breaks.
Exactly. It maps the exact base pair locations of the breaks across the entire genome. They took the neurons that had survived the tight squeeze and basically charted a map of where all the alarms had gone off.
And what they found is just a stunning piece of evolutionary architecture.
Yeah.
Because, you would assume that mechanical crushing would just cause random damage across the board.
Right.
But it's not random at all.
Not even slightly. The data showed the breaks were entirely strategic. They were almost entirely absent from active promoters and highly transcribed genes.
The vital stuff.
Yeah, the daily machinery that keeps the cell alive and functioning. Instead, the damage was heavily concentrated in transcriptionally inactive regions, specifically areas called Lamuna-associated domains, or LADs, and in repetitive retrotransposons.
The junk DNA.
Exactly.
Let me try a metaphor for this because spatial biology is hard to visualize. Imagine you have a shipping box,
Okay.
and inside you have highly fragile crystal glasses. Those are your vital active genes. To protect them, you pack them in the very dead center of the box, and you stuff all your useless, crumpled up junk mail around the outside perimeter, flush against the cardboard.
I like where this is going.
When the box gets crushed and kicked around in the mail, that outer perimeter of junk mail takes 100% of the physical friction and damage, keeping the core crystal completely intact.
That is a phenomenal way to picture it. Those lamina-associated domains, the junk mail, are regions of densely packed, silent DNA that sit right up against the inner wall of the nuclear envelope.
Ah, right against the edge.
Yes. When the nucleus gets squished through the pore, the greatest mechanical force and friction is absorbed by this dense, silent layer at the very edge. The molecular scissors are doing all their frantic cutting and jamming in the junk DNA, which perfectly shields the vital, life-sustaining coding regions safely tucked in the center.
It's just brilliant. The cell is willingly sacrificing the parts of its blueprint it doesn't use to save the parts it needs to survive.
Yeah.
But, as with any biological compromise, you have to wonder about the long-term bill.
For sure.
Even if you're taking damage in the junk mail, eventually the box is still taking a beating. What happens if the duct tape crew just doesn't show up?
To study the long-term cost of that, the researchers genetically engineered a special group of mice.
Okay.
They permanently deleted the gene responsible for creating that vital duct tape enzyme, the LIG4 gene. And they did it specifically in two ways. One mouse model, called Neurod1-CRE, had the gene deleted before the neurons started their massive migration journey.
Q4, okay.
And another, called Gabriel-6-CRE, had it deleted after they migrated.
Okay, so for the before group, the Neurod1-CRE mice, these cells are diving into the dense brain tissue, getting crushed, deploying the scissors, dissolving the scissors, and then they just have no tape to fix the gaping holes.
Nothing.
Do their brains just fail to form entirely? Do the cells undergo mass apoptosis and die?
That is the most shocking finding in the entire paper. You'd expect catastrophic failure, but no. The neurons survived.
Wait, really?
Yeah. When the researchers looked at the brains of these engineered mice under a microscope months later, anatomically, they looked completely fine. The layers of the cerebellum formed normally. The cells got to their destinations.
That the DNA breaks were still there.
Yes, because the duct tape was missing. The structural damage caused by the migration couldn't be repaired. These mice had permanent, persistent DNA damage for the rest of their lives.
That's insane.
In a normal mouse, the temporary damage is cleared within hours, but these neurons carried broken genetic strands forever.
Okay, there is no way a cell carries permanently severed DNA for its entire life cycle without a major consequence. I mean, even if it's just in the junk regions, damage is damage.
You're entirely right. It's not an immediate catastrophe. It's a slow burn. Over several months, as the mice aged, The researchers noticed they started developing mild but highly progressive motor deficits.
Motor deficits, like movement problem.
Right. They analyzed the mice's footprints and realized they were taking much shorter, hesitant strides. And when they challenged them to walk across a raised, narrow balance beam, the mice with the unrepaired DNA slipped far more frequently than normal mice.
So it wasn't a failure to build the brain, it was a failure to maintain it. Why does damage in the junk DNA eventually affect how a mouse walks, though?
Because a damaged foundation eventually creeps up into the house. When they sequenced the RNA to look at which genes were turning on and off, they found that those persistent unrepaired DNA breaks were causing chronic shifts in gene expression.
So the active genes did eventually get messed up.
Yes, crucial genes related to synaptic function and how neurons talk to each other started shutting down, while stress and immune response genes were constantly flaring up. The transcriptional changes strongly mirrored the genetic signatures we see in the very early stages of neurodegenerative diseases.
Wow. Let's recap this final piece because that is heavy.
Okay.
Bullet point 1. Failing to repair migration-induced DNA breaks doesn't actually kill the neuron.
Bullet point 2.
Yeah.
But it does leave lingering genomic instability, a persistent damage state.
And bullet point 3. Over a lifetime, this leads to mild motor decline and mirrors neurodegenerative aging.
Exactly.
So question for you listening. If normal brain wiring intrinsically creates genetic wear and tear, could some aspects of brain aging simply be the delayed bill for our brain's original construction?
It connects the very beginning of life to the very end of it in a deeply physical way.
It really does. Looking at the sheer scale of this, what Zhang and colleagues have illuminated is just terrifying biological resilience. To give you and me a functioning brain, billions of individual neurons have to survive a crushing, suffocating journey.
Yep.
They endure extreme physical distortion. They intentionally slice their own DNA to relieve the pressure. They cleverly absorb the hit in the silent regions of their genome, and they frantically tape it back together while on the move. And this chaos happens perfectly millions of times a second in the dark, silent spaces of every developing brain.
Which brings us to the most profound implication of this entire study, I think. We noted earlier that the repair mechanism, the NHEJ duct tape, is incredibly fast, but notoriously error-prone.
Right.
It frequently introduces tiny mutations, dropping or adding genetic letters at the break site.
Right, because it's prioritizing speed over accuracy. Just close the wound.
Exactly. Now, multiply that tiny margin of error across billions of migrating neurons, because billions of cells rely on this sloppy duct tape to survive their journey. It means that by the time a human brain is fully formed, the DNA sequence in one neuron is statistically likely to be slightly different from the DNA in the neuron sitting right next to it.
That is wild. Most of us are taught in high school biology that every cell in your body has the exact same identical copy of your DNA.
Right.
But you're saying the brain is actually a patchwork quilt of genetically distinct cells.
Yes, a phenomenon known as somatic mosaicism. And this leaves us with an incredibly provocative concept for you to explore. If every single neuron accumulates a slightly different set of microscopic mutations just from the violent act of migrating into place, could this mechanically induced damage be the hidden reason why every single human brain is so wonderfully uniquely wired?
Or conversely, could the tiny mistakes made by that duct tape crew during this perilous journey be the foundational origin of certain neurodevelopmental conditions that we currently can't explain with inherited genetics?
It's highly possible. The very damage that builds the structure might be the thing that introduces the variations that make our minds completely one of a kind.
The destruction is the design. Thank you for joining us on this deep dive. The next time you're out walking or thinking through a problem, take a moment to ponder the beautiful, violent, and incredibly resilient microscopic construction site that had to operate flawlessly just to give you the capacity to think.
A lot of grand pianos.
Billions of them. Think about those billions of grand pianos squeezed up the winding stairs, scratched, dented, but ultimately playing a perfect, completely unique melody. We will see you next time.